Hi everyone, it's my pleasure to give some updates on the OpenVaccine project today. So this has only been possible thanks to a lot of collaborative efforts of a lot of people, many of whom are here today. So this includes Eterna players, Eterna developers, and many researchers in Riju Das's lab and also our collaborator Maria Barna's lab at Stanford. So, as we all know, the COVID-19 pandemic has impacted all parts of our society and has caused immeasurable loss. Those of us who are fortunate enough to live in more vaccinated areas have recently felt the thrill of our lives reopening in some parts. But we also see, with the danger of the Delta variant, that this is really kind of a precarious freedom and it's threatened by new COVID variants, and there's still a race to be able to vaccinate more of the world before these variants mutate further. I'm showing here a New York Times headline from when RNA vaccines from Pfizer and from Moderna were first rolled out in the United States back in December 2020. And right alongside this headline, we see this worker packaging these vaccines using dry ice. And since the start of their rollout, there's been big questions about the stability and the shelf life of RNA vaccines. Medicines that require ultra-cold shipping are going to be limited to parts of the world that have the resources to manage these new ultra-cold shipping conditions and logistics. And so early on, we recognized that this fragility was going to exacerbate existing healthcare disparities between the global north and global south. But because these RNA therapeutics have a lot of potential, and since those of us present like thinking about RNA, we wanted to see if there was anything that we could do about this. And to get into the premise of how exactly we could help with OpenVaccine, first I want to talk about how conventional vaccines work and then how RNA vaccines work. So conventional vaccines are developed to create a weakened strain of a virus. This weakened strain can often take months to evolve and develop. Once it's created, it's that entire virus that is then in the vaccine that is injected. Your immune system then learns to recognize these weakened viruses to be ready for when the real thing might come along. The premise of RNA vaccines is to not inject an entire virus, but to create a part of the virus that is the part that the body is recognizing. So in the case of a coronavirus, it turns out that we can take just this spike protein, and if we can create a messenger RNA that codes for that spike protein and just inject that messenger RNA, our body's ribosomes, which we heard about earlier this weekend, can then create the spike protein on its own, and our immune system can learn to recognize the entire virus by just recognizing the spike protein. And so this really transformative technology, I just want to put up the names and faces of those in academia and industry who have been critical in making this happen and creating the vaccines that have reopened society for us. But RNA is an intrinsically unstable molecule. It's related to the reason RNA is not the molecule that is encoding our genes. So it's less stable than DNA. If it's subjected to heat or changes in the basicity of the solution, it can be prone to cut the message. And so this is a problem if your RNA is a message that is coding for a protein, because then you won't get the entire protein that you want to be presented to the body. So this is a problem, and what can we do about this? So one biological fact that we try to use against this is that when our friend the ribosome is reading this RNA and creating protein, there's a very complex code that it's following. And so you may have seen in a high school biology class one of these codon tables. So this is showing these 3 triplets of nucleic acids, each of which codes for an amino acid. I say high school biology, but also we see on Eterna, thanks to developers, we also have codon tables on Eterna. And so there's a huge amount of redundancy as to how many sequences can code for the same protein. And so, in fact, for the spike protein, that is the protein being produced by COVID-19 vaccines, there's a huge number of synonymous messenger RNAs that will all give us the same protein. So that's the first biological fact that we can try to use. The other biological fact is something that you're very familiar with as an Eterna player. And it's that most people are familiar with the idea that DNA base pairs, but RNA can base pair too. And there have been measurements showing that RNA that is base paired is more stable, more resistant to being cleaved than RNA that is single-stranded. So as just a little toy example, if I take this very short protein, if I went and took my codon table and enumerated all the possible mRNAs that code for this protein, there's about 25,000 of those. And so if I can think about one of these messenger RNAs and how likely it is to be cleaved by looking at the unpaired probability for any nucleotide. So if I go to a conventional server from a biotech company where one might traditionally go and design a messenger RNA, if I get back that sequence, here I'm showing the minimum free energy structure of it, but colored by this unpaired probability, you see that there's a lot of yellow regions. So a lot of regions that have a higher probability of being cleaved, but that in this stem area, the stark purple is predicted to be less degraded. So that's one sequence. If I go and enumerate through my 25,000 other sequences, it turns out I can find a sequence that codes for the same protein, but that is much more paired, and it turns out has roughly a twofold predicted increase in the shelf life for this little mini protein. So this is all well and easy enough to figure out the answer when there's only 25,000 mRNAs. Again, for our spike protein, which we want to be able to design stabilized RNAs for, here I'm showing the predicted structure of this spike protein mRNA as, again, coming from a conventional design source. And you see, again, that there's a lot of these yellow unpaired regions. So when we're presented with this astronomically number of mRNA vaccines, we had two big questions that we sought to answer. So one is just how much could we improve the stability of any given molecule this big? Is it still twofold like for the mini protein? Is it more or is it less? And then critically, if we could come up with a more stabilized messenger RNA, can it still produce protein that it needs to be able to be a vaccine and be useful in the body? So as you're all likely familiar with, when we have a problem in RNA design that we need creative solutions for, one of the best places to go to is Eterna. And I'm just not briefly going to skim over exactly how the open vaccine puzzles were created, but it was thanks to a huge amount of development by folks like John Nickel, LFP, many others here, to adapt the Eterna platform, as you have seen, to the open vaccine platform. And what we found is that for these full-length messenger RNAs, that we were predicted to see at least a twofold increase in the lifetimes for these. And so again, here on the left, I'm showing from the structure from the standard codon optimization, and then here on the right, a construct from Bugakman, which is predicted to have, I believe, a 2.5-fold increase in stability over this design. And furthermore, with our collaborators in the Barna lab, we were able to experimentally measure the lifetimes of not these full-length spike proteins just yet. The results are coming for those. But we have done many more studies on a shorter model protein, nanoluciferase. And so here I'm showing a time curve. So over a number of time points, folks in the Barna lab took measurements of how much RNA was remaining. And so we can see that for this designed superfolder construct from Eterna, that it has roughly a 2.5-fold increase in the lifetime over this less structured RNA. So this was very promising to see. Originally, we had just made these predictions using that model of P unpaired. So here again, I'm showing an arm of a messenger RNA colored by the P unpaired. But something that may jump out to you is that assuming that the degradation at a point in a hairpin and assuming that the degradation at a point in the multiloop seems like it doesn't exactly make sense. It seems like, that a hairpin might be more geometrically constrained or pulled tight to be able to let the backbone get more degraded more quickly than, say, something in a multiloop, which might be more protected. But there wasn't really the data to make any better of a model than the P unpaired model. So in order to do this, last year, alongside the OpenVaccine challenges, we also had launched this Roll Your Own Structure challenge, where we wanted players to create as many short sequences of diverse sequence and structure to then be able to collect data to degrade those and get a better sense of what type of RNA is more or less likely to be degraded. So I'm showing here a t-SNE plot of all the solutions. This is from the Roll Your Own Structure 1 challenge. Here are all the participants. I like this plot because you can see both that there's a lot of sequence diversity, but then you can also see clusters of sequences where players were drilling down on one particular hypothesis or another. So again, each of these points represents one of these different Roll Your Own Structure solutions. And Cladwang in the DOS lab then performed an experiment where all of these got assigned a barcode. They were subjected to these accelerated degradation conditions. And then thanks to this barcode, then all in one pot, we can assign little fragments of RNA back to the same construct. And it's then through this experimental process that we get this data that was then returned to players, where we can see for any particular nucleotide spot which regions got more or less degraded. And when we then take all this data and aggregate it, there are some really interesting things that jumped out to us. And I'm just going to show you next just one of these analyses. So here in these depictions, I'm going to be showing a backbone-centric view of the RNA. So because it's the backbone getting degraded, each little line here, so here I'm showing a 1/0 bulge, and the colored line represents that linkage on the backbone. So if I compare this 1/0 bulge to bulges where I'm increasing the length of the bulge on the 5' side, we saw, as we might predict, that as we increased it, we saw more degradation on the side that was being increased. If I do the same where I have a 0/1 bulge and I increase the 3' length, we also saw an increase in degradation on that side. Now, if I fill out the rest of the entries in this matrix, you might say, we might expect by the P unpaired model that all of these would be getting more degraded. But if we look at that, to our surprise, we saw that actually these symmetric loops down the diagonal of this stayed more protected, but that it was the more asymmetric internal loops that were more hydrolyzed. So we wanted to be able to take all these features and other features that players and that researchers saw and put them into a model that we could then turn into one number on Eterna. And our first pass at doing this, it was the DEG score model. So I created a simple machine learning model where I took as input features the sequence and then the secondary structure assignment for the sequences, and then based on a window of these values around every particular degradation site, so you used a linear regression to predict the degradation based on this window. And so we saw evidence in our tests that this performed better than the p-unpaired model. And so that was promising to us. I'm skipping over showing that data here. So I want to get to this next question, which is, so these superfolders, we showed in an experiment that they are actually more stable, but can they be translated? So I'm going to show you results from our experimental collaborators, again, for this model system of nanoluciferase. So after we had done experiments on roughly 200 RNAs, for these final experiments, we then selected 24 that were from Eterna players, that were from the algorithm ribotree, and that reflected a number of controls for standard methods for sequence design. So this bar plot is showing the in-solution half-life for each of these. So again, we can see roughly a twofold increase in the half-life of, the half-life being the time it takes for half of the RNA to degrade of the Eterna constructs and the ribotree constructs. We then took these 24 and compared their protein expression at 6 hours and then also at 24 hours. And we can see that the ribotree constructs and the Eterna constructs that actually produced more protein than the standard control that we were comparing to. So the fact that these superfolder constructs are within error and in some cases even more highly producing than the standard nanoluciferase messenger RNA was a surprise, and it was really promising to us to see this. And so it suggests that the most important factor for creating a messenger RNA that produces more protein might in fact be its stability. That a more stable messenger RNA will get to be around longer in the cell in order to produce the most protein. And there's a couple other interesting ribosome biology aspects to this too that would be fun to talk about in the discussion following this. So in conclusion then, thanks to Eterna player designs and experiments by our collaborators here at Stanford, we've shown that we can create messenger RNAs that are more stable and also produce more protein than standard messenger RNAs. So just as a final note, and as far as what's currently going on with open vaccine challenges, so at one point there was this round 7 where we asked players to create spike protein superfolders. And so while these constructs are much bigger and harder to test, I was personally just very struck by the diversity of sequences and structures that players were able to come up with. Because these sequences actually code for spike proteins, we wanted to make them publicly available for anyone engaging in RNA vaccine development to be able to use them. And so there's a repository online that I want to welcome you to go check out. It's DOSLab Superfolder COVID mRNA vaccines, where a good amount of information about these sequences and also just the sequences themselves are. Mark of how popular a repository is the number of stars it has. And you can see it has 47 stars, which I learned is actually a lot of stars for a repository. And so you are likely also aware that there's now another open vaccine round for the B1617 variant. And so I want to encourage everyone to Delta variant designs will also be put here, the winners, so that they will be made publicly available for anyone who a stabilized RNA sequence may be of help. And so with that, I want to again acknowledge all the people who have made this research possible, and I'm excited to talk more in the Q&A following this. Thanks very much.